All right, everyone. So super excited to introduce our keynote speaker, who is Dr. Marti Head. Welcome, Marti. Marti is an executive director of computational and data sciences at Amgen, where she leads a team applying mechanistic, machine learning, and AI computational methods to the understanding of disease biology and the discovery and design of biologic and synthetic therapeutics. Marti had a groundbreaking career where she worked in multiple different institutions before, including Oak Ridge National Laboratory, where she was director of computational biomedical initiatives and director of the Joint Institute of Biological Sciences. She also had spent 20 years in R&D at GlaxoSmithKline Pharmaceuticals, GSK, and many of those years she was the lead in GSK's U.S. Computational Chemistry team, whose accountability was to proactively and creatively apply all relevant computational tools to progressing drug discovery efforts from target selection through to selection of a candidate for clinical trials. Marti, we are very happy to have you here. We look forward to your talk. I'll stop sharing so you can start sharing your slides. You should be able to share now, Marti.
I am doing that even as we speak. And so
Wonderful.
Hopefully, excellent. And so it is a pleasure to be with all of you today. Thanks to Romy and Alex, Serene, and Alexei for the kind invitation. And Joni, I was excited to see that I was reading your mind just a little bit as I thought about the topics and the things I wanted to share with everyone today as we think about computer-aided drug discovery then and now. And so, let's see. And so, of late, we just see paper after paper after paper, press release about AI and drug discovery. It feels to me a little bit like we're not even at the top of the hype curve yet and still have well to go on the hype side. And, this is just a really, truly random selection of things that will show up in Google Scholar if you search for AI in drug discovery. I could have selected hundreds, if not thousands, more examples to flash on the screen to you. And really today, what I'd like to do is just pause and take a deep, calming breath and think about how we navigate that hype, how we move through it in order to identify ways to accelerate the hunt for safe and effective therapeutics that will make a difference for patients around the world. And so we're going to talk today about some of that prehistory, and then we're going to move on to thinking about the now, and then maybe a few thoughts about how we should approach the future from a philosophical viewpoint. And I hope that some of the things I share with you today will set up some of my friends and colleagues from the field for their talks in the coming 2 days. And so first—
Recording in progress.
First, I want to point out this paper from 1937 about the effect of structure on the reactions of organic compounds, especially benzene derivatives. This is the work of Lewis Hammett, looking at initially how as you change substituents at the meta and para positions, how does that shift the acidity of benzoic acids? And derived constants for those substituents that explained those relationships, and then learned from that those same substituent numbers translated to a whole range of reactions and basicities and many different other properties. And so I like to use this example to remind everyone that we have been doing machine learning since before there were machines to learn on. There is nothing new under the sun, and there are many things new under the sun. And so what is it that took us from doing machine learning on graph paper to where we are now? I'll also note the Metropolis algorithm that many of you on this call work with day in and day out, that existed in theory and in thought and in painstaking practice long before the coming of ENIAC and MANIAC, with which I'm showing you here, the leading cutting edge computer that was installed at Los Alamos during the war years. And the creation of that new computing architecture really enabled the ability to apply this algorithm and to do this work that leads to so much work in Markov chain Monte Carlo methods even now today. And so these are two concrete examples of the past of what we've done as computational chemists. I haven't even noted at this time quantum chemistry, molecular dynamics, neural nets, all concepts that were developed in those early days before the advent of computers, but then enabled just really a flourishing of new ideas and new thoughts and new creativity because of the existence of that hardware. And so here at the beginning of this talk, I'd like to offer just a few early reflections. One of those is that computational chemistry is a discipline with its own rich history, its own areas of research focus that are worthy of our minds and our attention, especially for those of us who have worked so many years in small molecule discovery. We can feel like the new kids on the block relative to organic synthetic chemistry that's been going on for centuries. But we are a discipline with a history, with a present, and with a future. I'd also like to point out that these new computational architectures opened up new ways of computing and even new ways of thinking. And finally, you may not have noticed in that picture before, but the two operators of MANIAC and 2 of the 5 authors on that Metropolis algorithm paper were women. Women have been in computational chemistry since the earliest days of computational chemistry. And that's true for cultural groups and multiple countries. We are a diverse community that comes together to do this work, to attack this discipline, and to make a difference. And so now we're going to transition a bit to what has changed between then and now. I don't think I'm going to steal any thunder from Stephen Burley, but I am going to use the PDB as my example on which to anchor this transition from then to now. And if you go to the RCSB website, you can always see this stats page, and we can see the phenomenal growth over the years of the number of crystal structures that are available to us to learn from, to train on, to use for answer fundamental questions about specific protein systems. And so I'm going to point out a few landmarks along this graph. And the first of those is this 1982 paper coming out of the labs of Tak Kuntz out in California at UCSF. And the thing that's amazing to me about this is that in 1982, Irwin and several other people that are familiar to many of us in this room were already thinking about protein ligand docking at a time when there were only 117 protein structures in the PDB, the early nascent PDB. And in addition to being one of the most gracious human beings I've ever had the opportunity to meet, Tak was also really prescient and ahead of the game on how these computing tools with this just very small amount of data at the time could lead to big things for the future. And then I'll go on to mention the CASP competitions, a large-scale experiment to proactively and predictively assess the performance of algorithms for predicting protein structures. This is the exact time period when I was finishing my PhD, thinking about geometric properties of protein structures down at Duke University with Michael Presant, and moving to the Center for Advanced Research for Biotechnology for my postdoc. And for those postdoc years, I sat outside the offices of John Molt and had the opportunity to learn from him and to see the way he thought about those proactive studies of what we're doing, of these models that we're building across the partition from Jan Pedersen as well. And really thinking ahead of time, how can we rigorously understand how our algorithms are truly performing? Moving forward in time, I will call out in the early 2000s work. I'm highlighting here the work that I was most directly associated with, but at the same time, our friends and colleagues like Kate and Georgia and Christopher and others at Merck were doing similar analyses and looking critically at how those docking algorithms that had been developed over the years from TAC's 1982 publication, how were they performing on the real world of drug discovery programs that we were doing in the pharmaceutical industry? How do we know which program to use? How do we know which predictions to take seriously and which to not? And so a contribution that I think we in the industry made to academia and to software developers in really having that kind of robust test on data that sat behind our proprietary barriers. And of course, that led through the NIH and the funding mechanisms there to first the CSER and then the D3R resource that made some of that data from inside of industry curated and available for making blind predictions for folks who were not inside our walls, to be able to expand that community of people who could do these rigorous evaluations of these tools that we were using. And of course, we have already heard and highlighted the work of AlphaFold in 2021, performing the best in that CASP competition that John had created all those years ago, creating the ability for us to learn from all of those structures all of the time, and to have this predictive prospective evaluation at hand so we could test out and see that step change in performance had happened, which then led, of course, to the Nobel Prize earlier this year. And one thing I would say to those of you at the start of your career now, looking at this history, looking at this amazing work that's being done by these Nobel laureates and others like them around the research community, don't worry, the work is not done. There is still much to do, even in understanding protein structure, even in understanding beyond protein structure to protein dynamics and the interactions of proteins with other things. There is still much for us to do. And so I'm going to take a pause here to offer a few more reflections based on that example of the work that has gone on over these years. First is that the PDB grew to its current size because of a steady accretion of data over time. If you look at that graph carefully, you will see that there is a roughly geometric progression in the growth of the number of structures that are deposited each year. But because this repository exists for us, that data has grown over time to be approaching 250,000 structures that are available now. Most of those structures were generated for a primary purpose. They were generated because someone had a specific interest in a particular protein for a specific disease indication, for a specific family of proteins that they wanted to understand. And here's where I call out some structures from the early days that some of you may remember, where there were literally hundreds of T4 lysozyme structures with small changes being made to create pockets in the interior of the protein. At one time, a big proportion of the structures in the PDB were of this one protein, with these structures that were created to ask questions of physical chemistry about the structures. And just calling that out because both the primary use and the potential secondary use of these structures are both important. And the next reflection that this led me to is thinking, as Joni mentioned, about the importance of data quantity, data quality, data integrity. This is where I call out pioneers like Helen Berman and Gary Gilliland, who sat at CARB with me all those years ago, and all of the rest of the PDB crew, who were prescient in creating a robust meta-ontology to describe protein structures, even back to the original PDB format that was trapped in a certain number of characters on each line, to the MMSIF formats that enabled more flexibility and reliability in that metadata, the tenacity in ensuring that they impose standards so that when you submit a structure to the PDB, it has that data quality baked into it, and hanging out that carrot of the publication publishers having the policy that you must submit and get a PDB code before you can publish the structure. That collection of things ensured that the data that is in there is reliable for us to use, to learn from, and even more importantly, the decisions sort of in the midlife of the PDB to ensure that structure factors were included as well, so that we can go back and refine structures that have questionable things and as methods of improvement. The final reflection that I want to offer about this example that I've shared with you is that each of those 250,000 structures is a dynamic-ish snapshot of a particular state of a particular protein construct. And in the case of X-rays, sitting in a particular protein crystal form. In the case of NMR structures, the challenges of having multiple models that map to the data that you see in the NMR experiment. And in cryo-EM, the temperatures and the matrices and the challenges of manipulating those images to generate a structure. It is all a snapshot that encapsulates some of the motion that we know is there, but is only one picture of what that structure truly is. And so this is my opportunity to remind all of us again that even wet lab experiments are a model of the thing that we really want to understand. We often cut off the ends and cut out the protein kinase domain without thinking about the other domains, or we think about them. Of course we do. We're careful scientists, but not having that data that necessarily tells us what's going on with those extra domains. And so, yes, a reminder for us all that even wet lab experiments are model systems of the biology that we care about that goes on in patients who have disease. And so now we're going to move on to, okay, so what has changed since then? We've talked about the growth of data. We talked about how computing was originally a transformative act that enabled new ways of computing and thinking. My father, many years ago, built our first computer, old 8088, sitting on a desktop that looked very much like this Heathkit here. And my phone, on which I'm keeping track of the time that I've been speaking, has more power than that computer that sat on the desk in our house in Florida. And over the years, even this traditional silicon-based computing architecture has grown to systems like the NVIDIA DGX boxes, and from my life at Oak Ridge, installing Frontier, the world's fastest supercomputer. Even within that current paradigm, there has been a huge growth in the computing capabilities that are available to us almost at the touch of a finger. And if you add in all the cloud computing resources that are available to us, we're in a rich time for having compute available to us. And as we think to the future, there are new architectures coming. Joni has already mentioned quantum computing. There's, of course, neuromorphic computing, where quantum computing takes physics as its model for how we might build a new architecture. Neuromorphic computing takes our current understanding of the brain as a way of thinking about compute architectures for the future. And there are untold insights and ideas and thoughts for the future that no one has even dreamt of yet, that open new possibilities for computing. And here I'd like to challenge all of you to think about, what is that new way of thinking that these new architectures can unleash? Not just how can I take classical algorithms and hopefully make them run faster on the very expensive, very energy hungry quantum computers that we're building, but how does that new way of moving from bits to qubits allow us to think about problems in a new way and develop new algorithms and new insights and achieve some of those translational visions that all of us have? And so we're going to roll back now to where we started this conversation today about AI for drug discovery. And I'm going to tell you a story from the second half of the last decade, from those early days when some of those publications started showing up in that rapid fire frenzy of publications that I shared with you early. And I want to think about using this example, not to say that it is the be all and end all, but to stop and say, what can we learn from the early era of AI and drug discovery that helps teach us what we need to do next to make sure that these methods are as impactful as possible? And so I'm going to talk to all of you about a public-private partnership that I was associated with both during my time at GSK and when I transitioned on to Oak Ridge National Laboratory, the ATOM Partnership, Accelerating Therapeutic Opportunities for Medicine, a public-private partnership under the auspices of then Vice President Biden's Cancer Moonshot. And this was a project aimed at creating the closed active learning loop, where we had property prediction coupled to design criteria, feeding into generative molecular design algorithms, and feeding that data into active learning mechanisms, so that when we see our predictive models drifting out of control, can we do designs to fill the gaps in the spaces we're computing against? Can we trigger designs that move us towards the molecules that we need to actually be drugs? And so many of you will have seen many versions of this kind of thing in the years since we began aggressively applying these kinds of techniques to drug discovery and computer-aided drug design. We did 2 pilot studies as part of this project. One, a historical example from aurora kinase B inhibitors at GSK. The second, selective H1 antagonists with partners at a small company in San Diego. Similar design criteria shared between the two, with some specific design criteria for each individual ones and specific qualities and quantities of data that we had to work with. And with these 2 things, we did a number of studies doing generative design, making molecules, testing it, and doing a prospective, predictive assessment, and using it and learning from it in action, building the plane while we're flying and learning what works while it's flying. And so what can we learn from that one early experiment in these kinds of methods? Well, the first thing we can and did learn is that generative design and multi-parameter optimization really does shift the distribution of both predicted and measured properties in the desired direction. And so just a couple of examples of graphs from papers from my colleagues in that time, looking at the aurora kinase example, where the training data and the first 6 months data that is measured is graphed here, and the best 250 over those iterations. You can see that we have moved the properties of selectivity in the direction that we wanted, and we have also begun shifting some of the properties, the other properties that go into making a drug that we care about as well. Other snapshots to let you see this in action as we go through iterations of the generative design algorithm, seeing the designed molecules pull away from the space of the ones that we started with, both there and in this picture. And so they absolutely take that whole distribution and move it in the right direction. However, it is absolutely true that specific predictions for specific molecules are very unlikely to be accurate. And so if we treat the number for solubility as a prediction of what the solubility will be, we're using it in the wrong way. It is not a prediction of the number. It is a prediction around that distribution and where it's heading. And so just a couple of snapshots. This is the predictive model for hERG, a typical experimental model for QT prolongation, which is the physiological observable that we really want to understand. And you can see that it has some signal there, but it is far from perfect. And some of our models are even worse. You know, there's sort of kind of some signal here, but there's a whole lot of molecules that are performing not particularly well. Some of those are at the bottom of the assay measurability, which may explain it. But nonetheless, it is clear that it is not a lot of signal that's showing up here. And these models are far from perfect, and we should not use them for single point predictions. But there's enough signal there to move that distribution in the direction that we want it to go, which highlights the essentiality of having that active learning loop and that tight coupling between dry lab and wet lab in the processes that we're building. And finally, just a thought that occurs to me from the time that I've watched this work going on. I think it is helpful if we think about some of these generative design algorithms by thinking about the language that is appropriate for the object we're designing and the task we're designing for. So let me tell you a little bit more about what I mean about that. Many people in this virtual room will be far more expert on this than I. So I'm going to just give you a somewhat high-level description. But in the early days, we were very much using variational autoencoders as our mechanism for doing the generative design and for navigating a continuous latent space. So we would take our model, we would encode it into that continuous space, and then decode it at the back end. I was very lazy when I was generating this graphic, just using the same molecule as the input and the output. But generally, you would get much different molecules as you navigated the space. And oftentimes, because small molecules are not off, let me step back and say this again. Molecules, in some sense, can be thought of as continuous because they clearly are a continuous distribution of electron density across a space that exists in the world. But they act as if they were atoms connected with bonds, with bond angles and bond rotations. And so a continuous representation of this organic chemistry view of a small molecule is a mismatch in language style and leads to us generating molecules that are sometimes crack, smoke, and ugly that chemists can't make. And so we spend a lot of energy needing to filter and apply other approaches to force our molecules that we design into reasonable chemical spaces. And so calling that out here, that we've drawn it as looking chemically reasonable, but that's not always how these algorithms work. But the developments in large language models for human languages alters an alternative path forward. And so we do have a language for describing small molecules. We actually have more than one, but this is SMILES, and this is a molecule simple enough that I could probably write this SMILE string by hand and understand it. But we can also describe more complicated molecules that are less simple to write by hand using this language. For those of you who are associated with Amgen, you may recognize this as Lumakras, our marketed KRAS inhibitor for certain kinds of cancers. And because we have this language, we can use large language model techniques as a core generative design module. We can mask parts of the structure. We can learn what transformations are possible. We can make predictions of a huge number of properties about those molecules. And we can generate new molecule designs. And so this approach offers an alternative path that I expect you'll hear more about as we go through these 2 days for thinking about how we do generative design of small molecule structures. These same approaches are equally applicable to large molecule therapeutics, to antibodies and protein therapeutics, and enzymes as therapeutics. There, the natural language that we're using is the language of amino acids, but the same kinds of strategies can apply, taking these models that were generated in a completely different space and applying them in a new and fresh way to the questions that we care about in drug discovery. And so just a few concluding reflections for all of us here, and then we'll turn it over to questions and conversations from the audience. So the first thing I want to say is, yes, there have been amazing innovations and successes based on using AI for in silico drug discovery. But if we rely on that alone, we will fail. It takes more than that to discover a drug that actually is safe and efficacious for our patients. And so we need to think about a holistic view on the computational side of the approaches that we apply. The informatics and analytics that puts data in front of the end user, allows them to make sense of it, begins to apply some algorithms to expose patterns in that data that let them make decisions based on the data they have at hand. And of course, predictive machine learning and generative artificial intelligence algorithms have an important and exciting role for us now and into the future as one of the tools we use. But we don't need to, we also shouldn't abandon the physics-based simulations that we have done for so many years that answer questions at a different level of detail, that provide us with mechanistic understanding, mechanistic hypotheses that let us probe the system in a different way. And finally, thinking about systems modeling. We heard Joni allude to this earlier as well. We have, since the late 80s, early 90s, as an industry, we have made a bet on rational design as the way to do drug discovery. We have made the assumption that a single molecule interacting with a single protein will have a beneficial impact on disease and on health. But we all know that the situation is much more complicated than that. We know that there are networks of proteins and cofactors and other biological factors that interact together in a very complicated network. And we need to be thinking about not just AI approaches to this, but a huge collection of approaches to understanding, if I impinge on these complicated systems in one or two or more places, how does that effect percolate through the system to have that impact on disease that we want to have? And so my final thoughts for you today, just reiterating again, if we rely on AI alone, we will fail. Drug discovery is hard. The things that we need to have in a single molecule to enable us to have a high probability that this molecule will be successful in getting to the site of action and having the impact that we need for efficacy, while also being safe and not having off-target effects or effects that are related to the target. And knowing that we can actually manufacture the molecule in sufficient quantities to make a difference. All of these things have to be in those special molecules that translate onward into the clinic. And for us to get there and to solve those hard problems, we require an integrated set of dry lab and wet lab tools to make progress. Finally, I hope you've heard a thread coming through in these remarks that as we carry out our computational experiments, we need to do so with rigor. Rigor in the way the data has been collected, rigor in the way the data has been organized and categorized and had the metadata placed around it, rigor in the way we approach our computations, and being very, very careful to not fool ourselves. Many of you have heard me say many times that I loved moving to computational chemistry because I never got tar in my test tube. My cultures never died. I never had, my experiment always worked unless the program crashed. And that's both a strength and a weakness. I always get an answer, but how do I know if that answer has any meaning for decision-making for drug discovery? That is the place where we have to be truly rigorous and not fool ourselves. And then from the beginning of my talk, I'd just like to reiterate that computational chemistry is a discipline. with its own rich history and its own exciting future, and that women and a diverse community of practitioners have been in this field for the earliest days and will be there on into the future as well. And so the body of work that I talked to you about today came from this set of organizations. And with that, Serene, I think I did okay on time. So
Perfect.
I am going to stop sharing and we can go to a Q&A session.
Thank you so much, Marty. How inspiring just to hear about like all your reflections. That was just amazing. And to hear a little bit about Atom as well, because I was an assay biologist at UCSF in Michelle Arkin's lab there, working with Atom. What a wonderful collaboration, as well as you talking about women being part of this. It's just extremely inspiring. So thank you so much for doing that. Lots of questions, Marty, so I'm just going to start tackling some of them. Thank you so much for everyone that asked questions. We're going to tackle as many as we can today, okay? So first questions we have here, has any study comprehensively compared all the experimental protein structures in the PDB to their AlphaFold models?
I feel certain that that work has been done, but I can't point to any specific example. As we have Stephen coming later on in this the series, I would definitely want to be asking him that question. I will say that in some of the, I don't actually work for a living so much anymore. I just talk to people for a living. But when I have gone back and looked to structures, they're close enough for doing some work with. And there are some questions, especially since the earliest versions of that algorithm didn't include cofactors, didn't include ligands that we know. For proteins that don't have structural models, there's clearly some things for us to work on there, and that's exciting. I'm sure there are studies like that. If anyone in the audience knows of some, feel free to drop them into the chat for folks to pay attention to.
Thank you, Marty. And because this is a discussion and I see Stephen on the line here, Stephen, would you like to add anything to that if you are listening? All right, we'll skip. Maybe we'll ask him next when he's presenting. All right, so the next question here is, what are your thoughts on what are the future trends in the in silico drug discovery? Is that quantum computing, metadynamics predictions, or AI methods like AlphaFold?
All of the above, which is a very lame answer, but it happens to be what I truly believe. I do think that quantum computing and thinking in qubits, maybe that opens up possibilities for us to think about the state function in a richer way, to think about the Boltzmann distribution of conformations in a different way, because we're thinking in qubits. I never want to miss a chance to say that there are alternatives to quantum computing that may be equally interesting and rich, and that may have a lower energy footprint than quantum computing. So yes, these new methods of computing, absolutely true. And yes, metadynamics and understanding what's going on with the dynamics of protein systems and how do those integrate into networks of proteins interacting with each other. I clearly believe that that's a place where we should pay attention to. But I'd really challenge all of you, as you think about the translational needs of our patients, what are the big open questions that are truly out there? And how can we think about knitting together these techniques and creating new ones that tackle those fundamental questions? That's the place where the Nobels for chemistry and medicine and physics are going to come in the future, is from thinking about it from that angle, and using these as tools.
Thank you so much, Marty. More questions, so let's tackle some more. How can we improve the validation of AI ML models?
I'm so excited that I can say I get to leave that to my colleague and friend, Pat Walters. He'll be talking to you tomorrow about that exact topic. I do know that I have some biases from my own past history, where a deep preference for fewer model parameters rather than more model parameters, an intense desire that we not overfit our models just because we have so much space and so much rope to hang ourselves with. And I think you will have heard a theme coming through completely around the need for prospective, blind challenges where we don't already know the answer, and we make bold and predictive predictions that we then go back and test. And finally, always having very carefully and well-chosen null models to which we compare. And so it is not enough to say, my new method does great on these systems. It needs to have error bars around that understanding, and it needs to compare to other much simpler models so that we know the true added value of the time and energy and intellect we're spending on that model. But I am going to stop there because I know that Pat will have an awful lot of really interesting and useful stuff to say about that tomorrow.
Thank you so much, Marty. That was very helpful. Thank you. Another question here: how the concept of generative molecular design is different from scaffold or atom hopping?
Interesting question. And so in some ways, it's not really that different. Just like I said, we've been doing machine learning since before there were machines to learn on. We've probably been doing generative design since before there were generative AI algorithms. And so in some ways, it's a new name for an old concept. Some of the new algorithmic things are innovations, but long ago, when I was doing calculations on a silicon graphics computer using Flow, QXP, this algorithm written in some of the oldest Fortran you've ever seen by Colin McMartin and Regine Bohacek, we had the ability to identify connection points and take a set of reagents and do the computational synthetic chemistry to explore what happens as to the binding pose and not great predicted binding affinity as you made those pairwise changes. Some of what's different is the increase in the amount of compute that we have available and the approaches for doing the multi-objective optimization, though those too have a precedent in the past work that we've done. And so this is the place where I stop and I say again, there is nothing new under the sun, but there's always innovation and growth and creativity for the future.
Thank you so much, Marty. We'll take one more question because there's so many. I don't want to bleed into the lunch, but I want to take at least one more question.
And I will just say that I'll stay on and at least type in a few answers to the questions that are in the chat before I log off and go find my lunch here.
That would be super nice of you. So we appreciate it. So here's one more question we'll answer live, and then Marty will try to answer as much as she can afterwards by typing. Where are we most significantly in need of more experimental data to train in silico models?
Biologics. Biologics. Back in my days at GSK, every molecule that was made and registered went through a set of standard assays. Those assays, as we've noted, are just model systems for the thing we care about. But it meant that over time, we had millions and millions and millions of data points in the same assay, run on the same robotics by the same operator over multiple years. And so, on the other hand, to create a biologic takes time and energy and expressing something in cells which can die on you. So biologics is a place that's on my mind. So another place that's on my mind is around, now that I have the privilege to work with the computational biology team that's in my department, thinking about what are the ways that let us tease apart biology in more physiologically relevant model systems, recognizing that those two are models, but that kind of data feels important to me. And really the data that would allow us to start thinking in this more systems level modeling perspective, that's a place that feels, another place that feels very rich to me.
Thank you so much, Marti. That was super helpful. Okay, so we are going to go to a lunch break now, and we'll be back at 12:45. Marti, again, thank you from all of us, the organizing committee, the attendees here. What a wonderful presentation. If you could just answer some of these questions, like you said, that would be fantastic. Everyone, thank you.